Hi, my name is Jacob Singer,
I'm Dylan Schuller.
My name is Payson Haran.
and we are Group 1. Today we will be presenting on XGBoost. XGBoost was developed in 2016 by Tianke Chen as part of his PhD work at the University of Washington, under the mentorship of Dr. Carlos Giestrin. The main goal was to create a faster and more scalable version of gradient boosting that could handle large datasets efficiently. What started as a research project quickly took off in the data science world. It was officially presented at the ACM SIG KDD conference in 2016. It also gained recognition at competitions like the Kaggle, largely because of its high performance, custability, and ability to handle missing values. The tool is fully open source and available under the Apache 2.0 license, which means anyone can use it, modify it, or contribute to it. It can find the full source code and documentation on GitHub. Currently, XGBoost is considered the dominant model for structured data analysis. It performs exceptionally well on datasets like spreadsheets, clinical records, and omics tables. Its ability to handle large, high-dimensional data with speed and accuracy makes it a go-to tool across many scientific and medical fields. In genomics, it's frequently used to classify gene expression and mutation profiles, helping scientists identify disease subtypes or predict treatment outcomes. It is also widely applied in disease risk prediction, using patient data to estimate the likelihood of conditions like heart disease or diabetes, offering outputs when paired with SHAP values. Currently, XGBoost has been adapted for drug discovery pipelines where it can help prioritize candidate compounds to predict drug target interactions based on biochemical data. XGBoost is flexible, efficient, and has made it a trusted tool across bioinformatics, healthcare, and pharmaceutical research. Looking ahead, XGBoost is expected to play a major role in AI-driven systems biology, integrating machine learning with biological modeling to uncover insights from complex datasets. For example, future applications may involve modeling disease progression or physiological responses using hybrid approaches that combine AI with dynamic systems modeling. We are also seeing a push towards integrative modeling, where XGBoost could help fuse multi-omics data like genomics, transcriptomics, and clinical variables to support precision medicine efforts. These applications require reliable models such as tools like SHAP that are critical for transparent feature attribution. XGBoost is also becoming more accessible through AutoML, which enables researchers to automate hyperparameter tuning and model selection, reducing the barrier to entry for non-programmers or clinical researchers. This slide provides a broad overview of the kinds of biological data that have been successfully analyzed using XGBoost across a range of biomedical applications. Starting with gene expression data, such as RNA sequence or microarrays, XGBoost is commonly used to classify cancer subtypes or distinguish between disease and healthy samples. Next, we have single nucleotide polymorphisms, or SNPs, which are used in genome-wide associated studies. DNA methylation is another area where XGBoost has shown value. It can detect epigenetic biomarkers for early cancer detection, especially from blood-based methylase signatures. In proteomics, XGBoost has been used to predict disease biomarkers and annotate protein functions. In the microbiome space, using 16S or metagenomic data, XGBoost can associate microbial community composition with disease states like inflammatory bowel disease, offering insights into host microbe interactions. Additionally, single-cell RNA sequence, the algorithm can classify different cell types and even track developmental trajectories, such as mapping immune cell states in tumor environments. When combined with clinical metadata like EHR data, XGBoost can power risk prediction models. In drug response data, XGBoost supports pharmacogenomic modeling by predicting IC50 values.
One of the most impactful biomedical applications of XGBoost is in cardiovascular health. In the UK Biobank study, researchers used it to predict myocardial infarction, or heart attacks, by analyzing a wide range of patient data, things like cholesterol levels, age, BMI, and even lifestyle habits. Because XGBoost handles missing values well and offers strong accuracy, it was a great fit for this kind of complex real-world dataset. It's also been used in cancer diagnostics, particularly in classifying tumor types from image-based or numerical data. For example, given a set of features extracted from a biopsy image, XGBoost can predict whether the tumor is likely benign or malignant. This has real potential to speed up triaging and assist doctors in making faster, more informed decisions. XGBoost isn't just for biology, it's used in many other fields too. In electrical engineering, it's been used to predict power system stability after a disturbance. In finance, it helps detect credit card fraud, calculate credit risk, and optimize investment portfolios. It's even found its way into agriculture, marketing, and cybersecurity. That versatility is one of the reasons it remains so widely adopted. There are three main ways to install XGBoost depending on your environment and needs. Option 1, pip install XGBoost. This is the easiest option if you're using a standard Python environment. Just run pip install XGBoost in your terminal or notebook, and you're ready to go. It's quick, simple, and great for most users. Option 2, Conda install. If you're using Anaconda or Miniconda, this is the recommended method. Conda handles dependencies better, especially across platforms, and the Conda Forge channel keeps things up to date. It's ideal for folks working in data science or with multiple packages installed. Option 3, build from source. If you want GPU acceleration or need more control over how XGBoost is compiled, you can build it from source. This is a bit more advanced and involves compiling the C++ backend, but it unlocks faster training, especially on large datasets. The official GitHub page has step-by-step instructions for this. For anyone new to XGBoost, we've provided a link to a short step-by-step tutorial video that walks you through installing the package and verifying that it's working. You can find the link to the video right here on the slide. After installing, it's a good idea to test your setup. Open a terminal or Python environment and try importing the library with import XGBoost. If there are no errors, you're good to go. For hands-on practice, we suggest trying to run a simple training example like fitting a basic classifier on dummy data. If you run into any issues or want more advanced configuration options like GPU support or custom builds, you can always refer to the official documentation or the GitHub repository, which are both linked here.
XGBoost is based on the concept of gradient boosting, which involves adding new trees, which is a structure used in decision-making processes, where each node represents a feature, branches denote decision rules, and leaves indicate the final outcomes or predictions. This enables the enhancement of the model's performance. Instead of fitting each tree to the original data, we fit it to the residuals, the errors made by the previous model. This approach allows each new tree to correct the mistakes of its predecessors. Moving on to loss function optimization. The objective at each step is to minimize a loss function. For classification tasks, we might employ metrics such as log loss, while for regression tasks, we often use root mean square error. Now moving on to additive model structure. XGBoost constructs the model additively, starting with a base prediction and then progressively adding small trees that refine the model towards the true value. The complete prediction function can be expressed as f of x equals f naught x plus f1 of x plus function f2 of x, and so on and so on. To prevent overfitting, XGBoost incorporates regularization by utilizing both L1 lasso and L2 ridge penalties to manage model complexities. This is one reason why XGBoost often excels even when dealing with noisier, messy datasets. First, I want to talk about error correcting trees. Training happens in stages with XGBoost. After the first model is built, the next tree doesn't start from scratch. It focuses specifically on correcting the errors made by the first one. This process continues with each tree learning from the residuals of the model before. It's like a group project where each person picks up where the last one left off, fixing mistakes and refining the results, kind of like this video. Furthermore, gradient descendant based loss minimization. The optimization is guided by gradient descent, but XGBoost takes it further by using both the gradient and the second derivative Hessian, and that extra info helps the model converge faster and more accurately. Moving on to ensemble of trees that improves accuracy. None of these trees are very powerful on their own. They are weak learners. However, when we add them all together, they become an incredibly accurate model.
Regularization is how XGBoost controls complexity and avoids overfitting. It's one of the key reasons the model performs so well even on noisy or high-dimensional datasets. There are three main types of regularization techniques built into XGBoost. First, we have L1 and L2 penalties, which come from traditional linear models. L1, also known as lasso, encourages sparsity by shrinking some feature weights all the way to zero, essentially removing them. L2, or ridge regularization, spreads the weight out and penalizes large coefficients. Together, they help reduce overfitting by keeping the model simpler and more generalizable. Next are parameters that directly control the complexity of individual trees. Max depth is a key one. It sets how deep each tree can grow. Shallow trees tend to generalize better. Min child weight is another one. It sets a threshold on how much data a node must contain before it can be split. This prevents the model from chasing small, potentially noisy patterns. Finally, we have boosting specific controls. The learning rate shrinks the impact of each tree. Lower values slow down learning, but often lead to better long-term performance. Subsample adds randomness by training each tree on a random subset of the data, which adds robustness and prevents overfitting. These parameters together give XGBoost flexibility to fit complex patterns while maintaining control so it doesn't overfit and stays reliable across different datasets. Here we're looking at how the model's performance improves over time, specifically by tracking log loss. Log loss is a great metric for probabilistic models because it penalizes confident but incorrect predictions. As we train more boosting rounds, we expect the log loss to steadily decrease if the model is learning efficiently. One of the most important things to check is the gap between the training and validation curves. If the model is generalizing well, the two curves should follow each other closely. If the gap is minimal, that means the model isn't just memorizing the training data, it's actually learning patterns that apply to new, unseen data. The stability of the curve suggests that hyperparameters like learning rate and max depth are well chosen. There's no sudden spike in validation loss, which would be a red flag for overfitting. If we wanted to be more efficient, we could also apply early stopping, ending training once the validation loss stops improving. When evaluating XGBoost, we often start with log loss, which penalizes incorrect predictions more when the model is confident but wrong. It's particularly useful when we care about the certainty of predictions. Another key metric is AUC, or area under the ROC curve. This measures how well the model distinguishes between classes across all thresholds, so it's helpful even when the dataset is imbalanced. And of course, we still consider accuracy, which tells us the overall percentage of correct predictions, but we always check class balance before relying on it too heavily. One of the nice things about XGBoost is its built-in feature importance scoring. The most common measure is gain, which tells us how much each feature contributes to improving accuracy when it's used in a tree split. This lets us rank features by how influential they are, and it's especially useful when we want to understand which variables are most predictive, like when gene expression values matter most in a biological model. To avoid overfitting and get a reliable estimate of model performance, we use cross-validation. This involves splitting the data into several folds, training on some and validating on the rest. K-fold cross-validation is popular, and we often use stratified versions to keep the class distribution balanced in each fold. It's also a key part of hyperparameter tuning, testing different settings and comparing results across folds.
Hello again. I'm Payson Haroon, and I'm going to walk us through our creation of a study titled XGBoost Model for Chronic Kidney Disease Diagnosis. CKD is often referred to as a silent killer because patients may not show symptoms until the disease is already advanced. By that point, options are limited and treatment is expensive. But if we can predict it early using routine clinical lab data, we not only improve patient outcomes, but also reduce costs by potentially millions per year. For this part of the XGBoost project, we set out to reproduce the core findings from this paper using the same dataset and methodology. XGBoost, as stated by my colleagues, is a high-performance gradient boosting algorithm that's especially good at structured data like this. Our primary goal was to validate the paper's results, specifically Table 7 from this paper, which compares model performance across different feature selection strategies. We wanted to see, could we match it, and if so, what was really driving that performance. Along the way, we also examined feature importance, looked at how sensitive the models were to losing those top features, and prepared the groundwork for our demo, where we move beyond the paper and test XGBoost in new, creative ways on the same dataset. The dataset we used and what this study uses comes from the UCI Machine Learning Repository, a well-known chronic kidney disease dataset with 400 total patients. Each patient has 24 input features and a label indicating whether or not they have chronic kidney disease. The complete table of features is available for viewing here. These features include values like age, albumin, bacteria, white blood cell count, hemoglobin, and many others. This is a mix of lab values, symptoms, and medical history. It is a good test case for structured tabular data. The prediction task here is binary classification. That is, predicting whether a patient has chronic kidney disease or not. So if they have a CKD class value of 1, it means that they are positive for CKD, and a class value of 0 means they are negative for CKD. Of course, this isn't a clean, perfect data set. It's small, it's imbalanced toward the CKD class, and it has missing values and categorical data. So we had to pre-process everything, including converting text like yes or no into machine-readable formats, and inputting missing values using means or modes depending on the column. So in short, this dataset is compact, noisy, and medically meaningful, perfect for showing off the strengths of a model like XGBoost. The original paper tested 5 different XGBoost models, each using a different subset of features. These included automated selection strategies like recursive feature elimination, embedded tree classifiers, and unsupervised selection, manual creation, and full feature model. In our reproduction, we matched those 5 models using the same feature sets described in the paper, and we used a shared stratified trained test split to ensure fairness across comparisons. Let me make this clear. We did not perform recursive feature elimination, embedded tree classifiers, and unsupervised selection. We simply copied the features stated in the paper in these feature lists shown here on the right. Each model was trained using the XGBoost package in Python, and we evaluated them using the same metrics as the paper: accuracy, precision, sensitivity, specificity, and mean absolute error, or MAE. This gave us a standardized way to see whether we were achieving similar predictive power and let us dig deeper into what was driving performance behind the scenes. Under the hood, we use scikit-learn for trained test splitting and evaluation metrics, and XGBoost for the actual training and predictions. And as you'll see on the next slide, our results were surprisingly close and a little surprising. On this slide, we're comparing the training dynamics of the full model and the RFE model across multiple metrics, including log loss and classification error, on training, validation, and test sets. Starting with the full model on the left, we can see that the log loss and classification error both decrease rapidly in the first 20 to 30 epochs and then flatten out. The gap between training and validation is minimal, which suggests the model isn't overfitting. Now, looking at the RFE model on the right, which uses only a subset of the most predictive features, we can see a very similar learning curve. Loss decreases smoothly, and classification error drops in the same pattern as in the study. What's interesting is that both models achieve very low test error, but the full model appears to generalize just a bit better on the validation sets. That may be due to the inclusion of secondary features that provide redundancy and help smooth out edge cases. Now here on this slide, we're comparing 2 additional models from the paper. The ETC model, which uses an embedded tree-based feature selection strategy, and the US model, which uses an unsupervised statistical method to select features, specifically the ANOVA test. Just like before, we're looking at both log loss and classification error over 100 training epochs for the training, validation, and test sets. Starting with the ETC model on the left, we again see very fast convergence in the first approximately 30 epochs with all 3 curves, train, validation, and test, closely tracking one another. This tells us that the model generalizes well and isn't just memorizing the training data. On the right, The US model shows nearly identical behavior, clean, stable convergence with low classification error across the board. One thing to highlight here is that despite using different subsets of features, both models achieve very similar training curves and generalization. This reinforces the idea that XGBoost is highly redundant. It can still perform well even when given different slices of the data, as long as the key predictive signals are still there. And again, we're seeing no signs of overfitting in either case. These learning curves are smooth, stable, and reliable. Now we're looking at our final model from the paper, the reduced model, which uses a hand-curated set of just 13 features selected based on domain knowledge. On the left side of the slide, you'll notice the now familiar pattern in both log loss and classification error. Again, we see rapid convergence in the first approximately 30 epochs, very tight alignment between training, validation, and test sets, and flat, low classification error on all three sets by the end of the training. What's most impressive here is that the reduced model performs nearly identical to the full model, despite using fewer features. This further supports our earlier observations that XGBoost extracts most of its predictive power from just a handful of key inputs, or features. Now on the right, we're showing ROC curves, one for the full model and one for the reduced model, for our results and the paper's results. These curves plot the true positive rate against the false positive rate across decision thresholds. The blue curve hugs the upper left corner in both cases, and the AUC score is 1.00, meaning both models achieve perfect or near perfect discrimination on the test set. So not only do these models converge quickly and generalize well, they're also extremely confident and accurate in their predictions. And again, it doesn't seem to matter whether you use 24 features or 13. XGBoost delivers. So far, every model we've tested, full, RFE, ETC, US, and reduced, has trained easily, converged quickly, and produced high-performance results on this small clinical dataset. That naturally raised the question, what's actually driving the performance? And that's where we go next. Here are our results after creating the 5 models from the paper. As you can see, all of the models performed very well, with accuracy scores in the 96 to 98% range. Precision and specificity were also consistently high. In most cases, we saw perfect or near perfect values. That means the models are not only accurate, but also very confident when they predict a patient doesn't have chronic kidney disease. One of the most interesting takeaways here is how similar all the models performed when we produced the paper, despite using different feature sets. This hinted to us that they were all leaning on the same core features, which we'll dig into the next slide. Overall, our reproduction was successful. The models trained with XGBoost matched or closely mirrored the performance reported in the original paper. So at this point, we were asking, why do all these models perform almost identically? The answer, as it turns out, and after many hours of attempted debugging, is that they're all relying on the same core set of features, mostly lab values that are biologically meaningful in the context of kidney disease. Across every model, whether it's the full model, the reduced model, or the ones based on feature sets from the paper like RFE, we consistently see the same top predictors. These predictors being hemoglobin, specific gravity, hypertension, packed cell volume, and albumin. These variables dominate the decision making in every model. So even if we reduce the total number of features, as long as we keep those few key ones, performance consistently stays high. That helps explain the minimal variation in model accuracy that we saw in the earlier slides. XGBoost is extremely good at zeroing in on strong signals, and in this dataset, those signals are very consistent. Now in terms of how our reproduction compares to the original study, we got very close, but there were a few important differences. We did not rerun the actual feature selection steps like RFE or ETC, we just used the feature list provided in the paper. We used a shared train test split across all models, which allowed us to directly compare their performance on the exact same dataset. This may have actually made our comparisons more fair than what the paper reported. And finally, we used a newer version of XGBoost because this paper was published in 2020, and we're doing this work in 2025. So some of the internal optimizations may differ from what the authors originally used. It's safe to presume that XGBoost is currently more powerful than it was in 2020. Still, the takeaway is the same. A handful of biological markers are doing all the work, and XGBoost is robust enough that model structure doesn't matter as long as those markers are present. Okay, now that we've reproduced the study, it's time to go deeper. For this next part, we'll shift into a hands-on tutorial where we use the same CKD dataset to show how XGBoost can be applied to explore new biological questions. This section is meant to serve as a getting started guide for using XGBoost with real clinical or biological data. We'll walk through a live notebook where we perform a feature ablation study where we progressively remove the most important features to see how much the model depends on them. We use an unsupervised learning with PCA and k-means to discover natural patient clusters and see whether they align with CKD diagnosis or suggest new subgroups. Then flip that into a supervised problem, training XGBoost to classify those emergent clusters. The diagram at the bottom shows the full pipeline we'll walk through. Start with the dataset, run feature ablation, train XGBoost, apply PCA clustering, then train again to classify clusters. This is really the heart of the tutorial, using machine learning not just to reproduce known outcomes, but to generate new insights and make predictions where the labels weren't already known. So with that, let's jump into the notebook. All right, let's get into the tutorial. We're working with the CKD dataset, or the Chronic Kidney Disease dataset, from the UCI Machine Learning Repository. And we'll start by loading and pre-processing it. But first, we got to do our imports. And that ran successfully. So now going into loading and pre-processing the CKD dataset. This dataset has 400 patients and 24 features, including things like hemoglobin, specific gravity, blood pressure, and a binary label, CKD or not CKD. This data is a little messy. It has a mix of numerical and categorical features, missing values, and some formatting quirks like yes or no strings. So in the first few cells, we clean that up, or the first cell. We replace missing values with means or modes, convert categories into numeric codes using factorization, and convert the target variable to integers so XGBoost can use it. Once that's done, we split the data into training and test sets using the train test split from scikit-learn. This pre-processing pipeline is foundational for any real-world biological data. You never get a clean CSV and go straight to modeling. These steps here mirror what you do in almost any health data science workflow. In this part of the tutorial, we're applying XGBoost directly to a real biological dataset to run a feature ablation study, where we systematically remove top-rank features to test how the model performs without them. We'll start by training an initial XGBoost model using this line. The full model equals XGBoost classifiers. A few things to point out here. We're using XGBoost classifier, the scikit-learn wrapper for XGBoost classification engine. N estimators equals 100. This tells it to build 100 decision trees. The eval metric equals log loss, and this sets the evaluation metric used during training. The random state equals 42, ensures reproducibility across runs. Then we'll fit the model using this code here. And once trained, we extract feature importance scores using these two lines here. This gives us a ranking on how much each feature contributed to decision making across all trees in the ensemble. From there, we perform the ablation. Starting with all features, we progressively remove the top 1, 2, up to 15 features and retrain the model each time. Now that happens in this loop right here, this for loop. We make no additional hyperparameter tuning here, just use a baseline configuration to test how robust the model is as we remove information. Finally, we evaluate each model using accuracy, the accuracy score, precision score, recall score, the ROC AUC score, and more, which gives us a full picture of model performance at each level of feature removal. So now we're going to run this cell. As you can see, XGBoost is very fast. Made 15 different models and trained them in about 1.5 seconds. These results are really interesting. It's remarkable how stable the scores are. Even when we drop 10 plus of the top features, this shows how well XGBoost handles redundancy and why it's such a good fit for messy biological data. That tells us that the model isn't overly reliant on any single input. It's using combinatorial logic across features, which is exactly what tree-based methods are good at. This kind of ablation analysis is super useful when you're working with expensive lab tests or limited data. Helps you identify what's essential versus what is extra. Specifically, what I found really interesting is that when the TOPS classifier hemo was removed, the accuracy slightly improved from the full model. Precision was not affected, but accuracy and sensitivity went up. And as you can see, these scores kind of start to progressively drop a little bit as more features are removed, although not very fast. And this speaks to XGBoost computing power. So we move into unsupervised learning, where we explore the structure of the chronic kidney disease dataset without relying on any class labels at all. We start by selecting a biologically relevant subset of features for clustering. Things like hemoglobin, albumin, red blood cell count, white blood cell count, appetite, things like that. And this feature selection is shown here. Then we apply principal component analysis, or PCA, using scikit-learn to reduce this 10-dimensional space into 2 principal components. That is shown here. This gives us a 2D representation of each patient based on the features we selected, and we can plot this to visualize the distribution of patients. Once we have that projection, we apply k-means clustering to try and identify natural subgroups. This line asks k-means to find 2 clusters based purely on the PCA reduced features, and it returns a label 0 or 1 for each patient, indicating which cluster they belong to. Now we'll run this notebook, and it comes up with this PCA plot. So this plot you see uses the labels to color the points, and it turns out the clustering actually aligns pretty well with CKD status, even though we didn't use the CKD label at all. We confirm that alignment by cross-tabulating the cluster assignments against the true class labels, and we'll show that a little bit later. One cluster was made up almost entirely of CKD positive patients, and the other had mostly non-CKD. So PCA and k-means uncovered biologically meaningful structure without any supervision. This kind of supervised analysis is valuable in biomedicine because it can reveal subtypes, mislabeling, or hidden patterns that don't show up when you rely only on class labels. After discovering meaningful clusters using PCA and k-means, we now flip the problem and treat it as a new supervised learning task. Can we train XGBoost to predict which cluster a patient belongs to based only on their clinical features? This is a useful trick in real-world biology. Once you've found patient subgroups, you want to be able to classify new patients into those groups without running clustering every single time. We reuse the same 10 features from clustering, hemo, SG, albumin, and so on, and split the dataset like this. Then we train a new XGBoost model using these 2 lines. And this is exactly the same structure as earlier. 100 boosted trees using log loss as our training metric with the random state 42. And once trained, we evaluate the classifier on the test set. So in our case, we achieved 97.5 accuracy, and the confusion matrix shows that it almost perfectly recovers the original cluster assignments, with very few mispredictions. That's a strong signal that the clusters weren't random. They're structured in the original features that XGBoost can learn, even though those clusters were derived without any supervision. So now we've gone full circle. We started with a labeled disease dataset, explored it without labels, found new patterns, and then trained XGBoost to learn those patterns and make predictions on new patients. The final section of the notebook, cluster composition versus CKD diagnosis, helps tie it all together. So on this line here, or these couple lines, shows how the unsupervised clusters align with the true CKD labels. One of the clusters contains almost all CKD positive patients, and the other contains mostly non CKD. So, even without labels, the structure of kidney disease emerged naturally in the PCA space, and XGBoost was able to recognize and predict the structure almost perfectly. This tells us two things. XGBoost isn't just powerful, it's interpretable and flexible across supervised and semi-supervised tasks. And two, the CKD dataset, though small, contains clear biological signals that can be discovered and modeled in multiple ways. And that's what makes this kind of workflow valuable. It's not just about getting high accuracy, it's about exploring and understanding the hidden logic of disease and using tools like XGBoost to make it actionable.
SHAP stands for Shapley Additive Explanations. It explains how each feature contributes to a machine learning model's prediction, assigning credit or blame based on cooperative game theory. We analyzed two figures from a paper titled XGBoost, a novel explainable AI technique in the prediction of myocardial infarction using SHAP model to predict myocardial dysfunction. Figure 1 shows SHAP values from an XGBoost model predicting myocardial infarction. Each row is a feature, for example, age and cholesterol, and each dot is a patient. SHAP values indicate how much that feature pushed the model toward or away from an MI prediction. Red indicates high values, blue, low. Figure 2 is a SHAP dependence plot for waist size and sex. It shows that increased waist size is associated with increased MI risk, especially for males, which is shown in red. Female individuals, shown in blue with similar waist sizes, have lower SHAP values, indicating reduced risk. Together, these plots demonstrate how XGBoost can provide interpretable predictions, helping identify key risk factors like waist size and sex in MI classification. First, one of XGBoost's biggest strengths is its speed and scalability. It's built-in optimized C++, which makes it incredibly fast, especially compared to many Python-based algorithms. It also supports parallelization, which means it can use multiple CPU cores during training. That's huge when working with large datasets, like those often found in biology. So whether you're analyzing thousands or millions of records, it handles them efficiently without slowing to a crawl. Another key feature is how it handles missing values. Instead of requiring us to fill in or drop missing data, XGBoost actually learns the best direction to take when a value is missing during tree construction. This is incredibly useful in biological and clinical datasets where missing data is common. For example, a patient record might be missing a lab result or a demographic field, but we don't have to throw out the sample or guess a placeholder. Finally, XGBoost is known for its accuracy. It consistently performs at or near the top in competitions and real-world applications. Part of that is due to its use of regularization, like L1 and L2 penalties, which help prevent overfitting. Even with default parameters, it does surprisingly well, but it's also very tunable if we want to push performance even further. That balance between strong baseline performance and deep customization is what makes it so powerful.
One of the first challenges with XGBoost is tuning. It comes with a lot of parameters, like learning rate, tree depth, and subsampling, each of which affects performance. While this gives flexibility, it also makes optimization tricky. Grid search or randomized search are often required, which can become computationally expensive and time-consuming. If you're not careful, you might end up with a suboptimal model just because tuning wasn't done thoroughly. In addition, less interpretable than linear models. While XGBoost delivers strong accuracy, it's also often considered a black box compared to linear models. Tools like SHAP or LIME can help bridge that gap, but it still adds complexity. In fields like healthcare or policy, where explainability is essential, this lack of transparency can become a barrier to adoption. The power of XGBoost can backfire on small datasets. It's so good at fitting patterns that it may also capture noise if there isn't enough data. Even though it includes regularization, overfitting is still a concern. That's why cross-validation is critical. Without it, you might think your model is doing well, when it's actually just memorizing quirks in the training data. Moving on to future applications of XGBoost. It has a broad use in clinical diagnostics. One of the most promising frontiers for XGBoost is its clinical diagnostics. As healthcare data becomes more digitized, we can use XGBoost to build models that predict risk for chronic diseases like diabetes, cardiovascular conditions, and cancer. These models can be integrated into electronic health record systems that provide real-time decision support, flagging high-risk patients for early intervention. In addition, we can integrate with multi-omics data. XGBoost is also well positioned to tackle the scale and complexity of omics data. Genomic, transcriptomic, and proteomic datasets are massive, and often sparse two things XGBoost handles well.
Thank you for listening to our project on XGBoost.